All right, here we are going to start moving very, very fast. So we are going to cover both 9.2 and 9.3. So this is glycolysis harvests chemical energy by oxidizing glucose to pyruvate. And after pyruvate is oxidized, the citric acid cycle completes the energy yielding oxidation of organic molecules. So again, this is what you need to know. I am about to go into extraordinary detail on at least many of these steps. You do not need to know that. If you memorize this chart, that is enough. Okay? So here we have glucose moving into pyruvate. This is a process called glycolysis. We get NADH and ATP out of it. Here we take that pyruvate, turn it into acetyl-CoA, that goes into the citric acid cycle. There we get more electrons, and we also get a little bit more ATP. Ultimately, all those electrons get put into the electron transport chain, and that produces most of our ATP. So let's start off here looking at glycolysis. What is it? So this is simply the process of splitting sugar into two pyruvate molecules. There are two phases to this. There's an energy investment and then an energy payoff. So we have to put a little bit in order to get out. This happens whether or not there's oxygen present at all. Okay, it doesn't need it. So we get glucose. We use ATP and put phosphates onto the molecule. Okay, that is the energy investment. And then we can start getting ATP out of it. So we put 2 in, we get 4 out. So we get a net gain of 2 ATP, but we also gain a couple of electrons across the way. Okay, this ultimately leads to pyruvate in water. So let's take a look at this. Here's a glucose molecule. We are going to add a phosphate group to it. So we're just going to pop it here on the end. And then we are going to rearrange it to make it look a little bit more symmetrical. So notice here, it looks a little bit more symmetrical. Why symmetrical? Well, remember we are going to be splitting this in two, so we need it to be even. If you take a look at this molecule, what is not even about it? Well, this group doesn't have a phosphate, this does. Boom, fixed. All right, so now we take this very symmetrical looking molecule and we can break it in two. Now, when we break it in two, it's not exactly even. It doesn't produce exactly the same two things, but we can interchange them, right? So we have an enzyme that can take this DHAP and turn it into GP3, which is what we want. Now, what I want you to remember is because this splits into two and then this gets turned back into G3P, we actually have two of these G3P molecules moving into the next phase. So here we have two NADs coming in, stripping off one electron from each one of these two molecules, and this produces a reduced form of NAD. This is the resulting molecules from that, and remember we have two of them. We can then take this phosphate and pop it back onto ATP. Here we have a phosphate group attached to a carbonyl. That is not stable. We can utilize that to drive the generation of ATP. We now have two molecules of this. We can then subtly rearrange it, take a water molecule off, and produce another high energy compound. So here we have a phosphate group attached to a carbon that has a double bond. That is also an unstable scenario. It wants to get rid of that phosphate. We can utilize that to create another set of ATP. And we're getting two because we have two of these molecules. It's happening each one, right? And what we end up with is two molecules of pyruvate. So the energy investment phase, we take glucose, we add phosphate onto it, we rearrange it to make it look symmetrical, we add another phosphate to make it even more symmetrical, then we split it into two. When we do that, we'll take the G3P and convert this into G3P. We now have two of these molecules moving into the next phase, where we can harvest a couple of electrons, producing an unstable compound. We can then take that phosphate and pop it back onto ADP to produce ATP. We do a little bit more rearrangement and again end up with another unstable compound. We can then pop that phosphate group off, leaving us with pyruvate. And that moves us through glycolysis. This all happens in the cytoplasm. We've generated ATP and also electrons. It now gets imported into the mitochondria, and there it goes through a process that we call pyruvate oxidation to produce acetyl-CoA. This gets dumped into the citric acid cycle, where we finish off gleaning all the electrons and make a little bit more ATP. This does not work if you do not have oxygen. Okay, so in the absence of oxygen, this whole entire process just ceases to function. So let's take a look at this pyruvate oxidation. So this is a process where we're going to take pyruvate here, and we are going to import it into the mitochondria. In the process, we're going to pop off one of this carboxylic acid groups as a carbon dioxide, because we don't need all three carbons, we just need two. Those two left, we then strip off a couple of electrons, and that process of stripping off electrons helps facilitate us putting it on to a molecule called coenzyme A. This produces acetyl-CoA. This group here is also known as an acetyl group. One of the groups that I didn't force you to memorize is just a combination of a carbonyl with a methyl group. Now that we have acetyl-CoA, we can now feed acetyl-CoA into something that we call the citric acid cycle. Here we are going to slowly bleed off some carbon dioxide. We are going to glean a bunch of electrons off of it. You are going to generate a little ATP along the way, and then pull off even a couple more electrons. So here we start off with pyruvate, which is a 3-carbon molecule. We pop one of those carbons off. We now have acetyl-CoA. This is what we just talked about. This is then going to go into the citric acid cycle. We're going to generate 1 ATP, 3 NADHs, and 1 FAD, which is an equivalent to NAD, per turn. And remember that we have 2 of these molecules going in, so we're going to do a couple of turns. So acetyl-CoA, the carbons are marked in red, we essentially transfer those carbons onto something called oxaloacetate. Here, some of the carbons are marked in blue, some of them are in black. So we now produce this molecule called citrate. We can then rearrange that to produce isocitrate. Isocitrate, we can then glean a couple of electrons off. That is going to liberate another carbon dioxide. So you'll notice that here we have this carboxylic acid group at the end. This one is now missing. That is what popped off. In the process of popping off, we kind of had to rearrange these bonds, and now instead of a hydroxyl, we have a carbonyl. This is alpha-ketoglutarate. We then do this process again, gleaning another carbon dioxide and gaining a couple more electrons. In this process, we also are able to pop it back on to a coenzyme A molecule. That coenzyme A is then going to take that succinate group and liberate it off into its own thing. That has a sufficient amount of energy to drive the formation of ATP through an indirect process where we form GTP, and then GTP can make ATP. Not important, right? We make ATP here. Now we can take this molecule here, and then we can take a couple more electrons off, The result of that is instead of having a single bond here, we now have a double bond, because since we've stolen some of the electrons and some of the hydrogens, it has to rebond with itself to fill the missing gap. That then gets converted into malate, which we can then strip off a couple more electrons in order to produce oxaloacetate, which was the beginning molecule of this cycle. Hence, it is in fact a cycle, and the end product is one of the reactants of the beginning part. So as long as we have acetyl-CoA coming in, we are good to continue the cycle. So this is the overall process that we looked at: glycolysis, glucose in the pyruvate, we generate ATP, we get some electrons. And then if we have oxygen present, we can import pyruvate from the cytosol into the mitochondria, turn it into acetyl-CoA through this process called pyruvate oxidation. We liberate carbon dioxide here. This acetyl-CoA gets put into the cycle, which turns around. We gain a lot of electrons, and we also generate more ATP. Ultimately, all of these electrons are then going to be put into the electron transport chain and used to generate ATP. So again, I gave you a lot of detail. You don't need to know it. Memorize this figure. If you understand this, you're good. If you are planning on progressing on in biology, you may stop and linger on some of the chemical logic that is happening within these steps. Another big part of the citric acid cycle is it produced a lot of complex molecules throughout the cycle. A lot of those molecules are actually precursors to other things, such as various amino acids. So sometimes we will take compounds out of the cycle and turn it into an amino acid. Or conversely, if we take an amino acid, we might be able to convert it into one of these compounds, feeding it in. Okay? So this is another way that energy can come into this kind of what we call a central pathway. Acetyl-CoA is another common entry point. So when we take fat and break it down, we actually break it down into 2-carbon acetyl groups. That is consequently also how we built the fat to begin with, 2 carbons at a time, through this acetyl-CoA entity. So consequently, fats can feed into here. They can also feed out of here. All right, so again, concentrate on this figure, but I did give you a lot of information. We're then going to explore oxidative phosphorylation in a bit more detail. And if you don't believe that this is a brief overview, I have and will do again a 2-hour lecture on glycolysis alone, a 2-hour lecture on pyruvate oxidation and citric acid cycle. So this is in fact a huge condensation.